Hi everyone, I'm Niloufar Nourmohammadi. I'm excited to share my research with you in Alternative Protein Week, and I'm truly grateful for this opportunity. I have a background in food science, holding a master's degree from the University of Idaho, and my research has been focused on exploring innovative approaches for alternative protein and their modification. Now, let's get started. Well, in recent years, as the demand for alternative proteins, specifically plant proteins, has risen, one of the key challenges has been improving the digestibility and overall consumer experience of plant-based products. And my research also tried to address this challenge. Today, I'll be presenting on improving digestibility of pea-based nugget through enzymatic hydrolysis. In this research, I explored how enzymatic techniques can enhance the digestibility and some other physicochemical properties of pea protein as a key ingredient in many plant-based products. Now, let me guide you through the presentation. So, I'll begin with the motivation and background behind this study, why we need to do modification, why there's a need for that, and what are the techniques that we can use for it. And after that, we'll move on the protein modification, the process that I'll explain the enzymatic technique that we use to improve the digestibility and some other physicochemical properties. And after that, I'll share the results of this experiment, followed by a product development section where we apply these findings in a real world application. And finally, I wrap up with the conclusion and discussing the future impact of this research. Now, let's begin with the motivation and background behind this study. As many of you are aware, there has been a noticeable global shift towards plant proteins. There are two major factors behind this transition. One of them is better sustainability of plant-based proteins, plant-based sources, and the other one is some health concerns regarding the animal sources. Main sources of plant proteins are like legumes, grains, seeds, nuts, and algae. Although they have their own nutritional value, when it comes to incorporating into the food matrix, they pose some significant challenges, specifically in replicating the general eating experience compared to the animal sources, like the texture, the flavor, like that general experience. There are some major problems with plant proteins, like challenges. One of them is limited functionality of plant proteins. So they cannot often provide the same functionality compared to animal sources. And regarding to understand that, we need to understand the complexity of protein, which is really important, is a really key factor. So proteins are complex biomolecules, and there are some factors that can affect their functionality. There are some intrinsic factors like the sequence of amino acid and that three-dimensional conformation that it has, and some extrinsic factors like the pH and like the temperature of the environment. Besides that, there is a huge challenge with its digestibility because of the presence of anti-nutritional factors. So these naturally occurring compounds, like for example phenolic compounds, they can interfere with the function of digestive enzymes like trypsin, like pepsin, and this leads to a reduced ability for body to efficiently break down and absorb plant proteins. So researchers are trying to address these challenges, and there are some ways in order to modify protein in order to improve these challenges. So, there are three categories of modifications, like physical, chemical, and enzymatic ones, like biochemical ones. And during the presentation in further slides, we will cover them. When it comes to modifying methods, there are three main categories: physical methods, chemical, and biochemical methods. In physical methods, There are some main methods like applying heating or applying shear and force using ultrasound or doing thermomechanical processes, which is a combination of applying heat and shear force simultaneously. They are usually cost effective and there is more consumer acceptance towards them. When it comes to chemical methods, it's usually about introducing a new compound or molecule which leads to the change and alteration in the function and structure of your target molecule. Chemical methods usually have high specificity and efficiency, they're low in cost, and they're more user friendly. So in this category, we have the use of chemical agents like salts or like doing alterations in the pH. So extreme pH levels can trigger the unwinding of polypeptide chains, leading to subsequent folding and reorganizing of the protein when we neutralize the pH. And it can cause the exposure of the buried functional groups of the protein and can alter the functionality of the protein. And last category that we are going to talk about is biochemical ones. It's usually about using enzymes. It needs a milder reaction environment compared to like chemical ones or like physical ones, and it's easier to handle compared to the chemical ones, and easier to control it. There are different enzymes that we can work with it, but they're usually fall into two subcategories: enzymes that doing hydrolysis or enzymes that doing cross-linking. Regarding the enzymes doing hydrolysis, we have pepsin, we have trypsin, we have alkylase. And regarding the enzymes that doing cross-linking, we can call the enzyme transglutaminase. So the whole idea behind these different modifying methods is about changing the structure and the conformation of the protein and changing the exposure of the functional groups, which can finally lead into the different functionality and different behavior of the protein. In the next few slides and this slide, I try to simply demonstrate just some of the ways in different categories that you can use to do modification. Regarding the physical modification, you can apply thermal or mechanical treatment by just simply using a water bath in order to apply thermal treatment, or like using a rheometer to apply shear force, or like using extrusion that in a controlled environment you can apply thermal and shear force treatment in order to change the conformation of the protein. And by doing that, you technically change the form of the protein, and there's a possibility of exposure of the functional groups of protein, which are usually buried in the core of the protein. But by unfolding that, you are altering that structure, and that can lead into the alteration in the functionality of the protein. So the picture in this slide is just simply trying to show the function of like an endoprotease in doing hydrolysis. So as I've mentioned, we have enzymes doing hydrolysis or doing cross-linking. So we can see hydrolysis in this picture. So as an endoprotease, like like alkylase, which cleave the peptide bonds within the protein, and it can provide like smaller fractions of protein at the end. So in that case, technically, you're going to change the structure of the protein, and there's a possibility of exposure of different like functional groups that they were not exposed before, and you will see a different behavior of protein after doing the hydrolysis. And this slide is talking about using binding agents as a part of like a chemical type of modification in order to improving the crosslinks within the protein structure, which can affect like the gel strength that is prepared by that protein. So there are different types of interactions. We have covalent and non-covalent interactions. And studies showed that the non-covalent interactions, like the hydrogen bonds, hydrophobic bonds, and electrostatic interactions, they have a main impact in determining the strength of the gel. You can use different agents like using ions, like using calcium ion, or using different agents in order to boost these interactions and modify it and like improving the strength of the gel, the protein. So, considering the challenges that you're facing in order to use a plant protein, like pea protein, and the ways that we can try to modify it and to address this challenge, we hypothesize that by applying enzymatic treatment, doing hydrolysis, as well as adding some sort of binding agents, we can improve the digestibility of pea protein, but maintaining the gelling capacity of the protein. And the objective of this study was to develop a plant-based nugget, a pea-based nugget, with enhanced digestibility while maintaining the texture compared to the control, which is a pea nugget without doing hydrolysis. So the modification step in this experiment overall included adding pea protein isolate powder into different levels of calcium chloride, like 4 levels of calcium chloride solutions containing carboxymethylcellulose as an enhancer for the strength of the gel. And then at the optimum temperature and optimum situation, the enzyme was added for a specific time, and after doing this hydrolysis for that specific time, the suspensions were heated for 10 minutes at 85 centigrade, and for like the activation, and then for the gel setting, and then after that, cooled down to 4 centigrade for an overnight, and then after the overnight, we tested the gels for its strength and for the digestibility and other parameters that I'm going to share some of them with you. So in this slide, the picture A is showing the difference in the degree of hydrolysis at different levels of binding agent, which is calcium chloride here. And pictures B and C are showing the difference in molecular weight distributions at different levels of calcium chloride after and before doing hydrolysis. So the degree of hydrolysis has been checked by the alpha-aldehyde method, and we can see that by the increase in the level of calcium chloride, we have a decrease in the amount of hydrolyzed protein, which can be explained by the fact that the binding agents can cover the sites that enzyme can work on it. But generally speaking, compared to the control one, we have a higher level of hydrolyzed proteins after applying enzymatic treatments. And regarding the molecular weight distribution, we can see thicker and more bands in the lower molecular weight fractions level compared to the time that we don't apply enzymatic treatment. And technically, we can see some of the bands for like ligament, vesseling, convesseling, and lipoxygenase technically disappeared. But regarding the different levels of calcium chloride, there were no significant difference between them. So enzymatic treatment has been applied, and we saw that we have a higher amount of small fractions of proteins, and the structure of protein has been changed. But in order to see what is the change in the rheological properties of the gel after doing hydrolysis, we monitored the storage modulus G prime as an indicator of the rheological properties of the gel. So in this slide, you can see a part of rheological studies that we've done on the gel. In the pictures A and B, there are results of the amplitude sweep test and on the pea protein and hydrolyzed pea protein. And we can see after doing hydrolysis, generally speaking, we have lower amount of G prime, which is an indicator of the strength of the gel. But after introducing the calcium chloride, we can see an increase in the amount of G prime and the gel is getting stronger. It can be explained by the fact that there are more electrostatic interactions that involve into the gel matrix. So in the picture C and D, we can see the results of another test, creep recovery test, which is about applying the force axially on the gel and checking the permanent deformations of the gel and the strain. And after doing hydrolysis in picture D compared to C, we can see we have larger amounts of strains and permanent deformations. But after introducing calcium chloride, we can see the amount of strain decrease and the gel is showing more rubbery structure, but up to an optimum level. Even for the amplitude sweep, we have an optimum level for the increase in the strength, and after that, we may have deterioration. In this slide, you can see the results of the digestibility experiment and how the digestibility of protein changed after doing enzymatic treatment and after the digestion process. So, generally speaking, in picture A, you can see after enzymatic treatment, you have a higher level of degree of hydrolysis after digestion, which can be explained by the fact that you have more exposed surface for the digestive protease to work on it after treating with enzyme. And following that, you have more soluble proteins in your system and after the digestion. But after introducing the calcium chloride as a binding agent, because of more formation of larger aggregates, you can see a decrease in the amount of degree of hydrolysis and soluble protein content. But when you compare the optimum level of calcium chloride and doing hydrolysis compared to the control one, you can see a significant improvement in the amount of degree of hydrolysis and in the amount of soluble protein content after digestion. So in previous experiment, we saw that there is a potential to improve digestibility, but kind of maintaining the texture. So in order to see the application of that in a real world product, we developed a pea-based nugget. You can see the pictures of these nuggets. The nugget made by pea protein on the right side and with the hydrolyzed pea protein on the left side for texturization, a twin screw extrusion has been used. And the formulation is saying the only difference is the form of the pea protein. One of them is hydrolyzed and the other one is not. And the amount of calcium chloride has been chosen based on the results that we saw, and we got the optimal one to use that. So in the next slides, I'm going to share some of the results of the assessments that we had on the pea protein to see what's going to be the difference in its texture and also in its digestibility as the key factors that we wanted to see how the enzymatic treatment is going to work on it. So we want to try by developing a plant-based product. The design is to mimic the animal-based one and trying to replicate the same eating experience for the consumer. One of those parameters that can be determining in this assessment is texture profile analysis, the texture of the nugget to see how it's changed after doing enzymatic treatment. So in this slide, you can see the results of texture analysis and the parameters hardness, cohesiveness, springiness, and chewiness has been checked and compared between nugget made with pea protein and nugget made with hydrolyzed pea protein. And surprisingly, there were no significant difference between them, and that's very interesting. So in that case, it seems like we were able to maintain the texture of the nugget, but we want to see if we had improvement in digestibility as well or not. And now in this slide, you can see the results of the in vitro digestibility experiment for the nuggets. and comparing the nuggets made with pea protein and hydrolyzed pea protein in regards of their degree of hydrolysis and soluble protein content. And surprisingly, we had about 25% improvement of the um, in the soluble protein content and degree of hydrolysis, which is truly interesting. So in that case, we were able to see a significant improvement in digestibility, but no significant difference between the texture, which is interesting. So as a conclusion, we could see that the incorporation of calcium chloride as a binding agent into the pea protein gel after doing hydrolysis has a significant impact in improving the gelling capacity. The importance of that is that we can improve the digestibility of the plant protein, but also maintaining the texture and not deteriorating the experience of eating of the plant-based product for the consumer. And that can open doors to the use of plant protein gels with favorable viscoelastic properties, but a better digestibility for future use. So part of the presented work has been published in a peer-reviewed journal, Food Research International in 2024. And if you're interested in more details, I recommend you guys to take a look at the paper. It's going to be interesting. And last but not least, I would like to thank my advisor and co-advisors, Dr. Chen, Dr. Campanella, and Dr. Rizaman for their guidance and support. And this project has been funded by GFI, and I'm truly grateful for this opportunity. Well, I hope these insights have been useful, and thank you for listening to my presentation. And please do not hesitate to reach out to me if you have any further questions. You can see my contact information in this slide. Thank you.